All right, let's go on and we'll talk about the gram-positive aerobic rods or gram-positive bacilli. Corynebacterium diphtheriae, Listeria monocytogenes, and Bacillus anthrax. Notice I did not put other species in here. We're just going to talk about those specific organisms. Corynebacterium diphtheriae does have metachromatic granules. Remember we talked about that earlier. It is non-motile, catalase positive. It is the diphtheroid Corynebacteria species that we see in sputum that has been contaminated with saliva. Listeria monocytogenes is the gram-positive rod. It loves the cold. All right. What are some of the buzzwords with it? Tumbling motility at room temperature and umbrella motility at room temperature. Also, look at the one characteristic that separates this baby from group B strep, and that is the catalase test. Because many of the tests, biochemical tests that are positive for group B strep are also positive for Listeria, with the exception of the catalase test. All right, so if it looks like group B, remember camp test positive, sodium hippurate positive, catalase positive should lead you to Listeria monocytogenes. Now, Bacillus anthrax, some of the buzzwords that go with Bacillus anthrax are string of pearls with penicillin and the Medusa head colony that Bacillus anthrax makes. Here's an example of Bacillus anthrax. Remember, all Bacillus species make spores. Mycobacterium are the acid-fast group of organisms. You use the Ziehl-Neelsen acid-fast stain. If you gram stain them, they will look gram-positive. You'll see like beads of gram-positive organisms in a sputum, which would lead you to say, let's go ahead and do a acid-fast stain. Here is your Ziehl-Neelsen. This is not the Ziehl-Neelsen. This is actually the auramine-rhodamine fluorescent stain. Some of you did get to do this in microbiology. It is now one of the standards for acid-fast testing. You must do the fluorescent stain, and then many labs will also do the acid-fast Ziehl-Neelsen stain. For Mycobacterium, you need to digest the sample using some sort of acid. Here, N-acetyl-L-cysteine sodium hydroxide is used. That kills all of the extraneous organisms except for the very waxy walled organism of Mycobacterium. You put it on Lowenstein-Jensen or Middlebrook, which is a media specifically for cultivation of Mycobacterium, because you want to grow it for a long period of time. Now, Mycobacterium tuberculosis has a couple buzz things that go along with it, is that it is cauliflower-like. It is a slow grower, 2 to 3 weeks, and it is niacin positive. Those are, if you don't remember anything else, just remember those couple things about Mycobacterium tuberculosis. Notice it does look like a little cauliflower here. You see that? Actinomycetes israelii is an anaerobic organism that gives you what is called the molar tooth colonies. This also gives you what is the sulfur granules in drainage. Think of the tooth abscess, that you look at the abscess and you have big old granules, or looks like you have sulfur granules in there, and when you smash them, you'll see that it is the actual organism that is inside these hard encased little shells. All right. Nocardia, partially acid fast. If you see a buzzword, Anywhere that says partially acid-fast, always think of Nocardia. Now, Haemophilus. Haemophilus influenza is a gram-negative pleomorphic cocobacilli. The characteristics with Haemophilus are that it needs X and V factor. Your X factor is hemin, your V is NAD. Each Haemophilus species has different requirements for X and V. Now, your chocolate contains X and V, and if you see any test question that says this organism is satelliting on a blood agar plate, immediately think of Haemophilus. So remember, the buzzword satellite phenomena is going to go along with Haemophilus. Where you'll have a problem if there's more than one genus listed of Haemophilus. But at least know you could get it down to 50% if you see this word. Okay? Now, Haemophilus influenza, we know, causes meningitis. Used to be the #1 cause of meningitis in children. With the Hib vaccine, we're seeing those statistics go down. There is a vaccine for Haemophilus influenza type B. It needs both X and V factor. You can also do some antigen testing. So Haemophilus influenza needs X and V factor. Here's your satelliting. The Staphylococcus aureus organism is made in an X, and then you'll see around the streak, or if you're just using a regular culture where you'll have staph as normal flora, you'll see the satelliting of Haemophilus. I bet you most of you have seen that. The tech maybe said, here, take a look at this. Do you see what's on here? Because it's really kind of neat to see. Haemophilus ducreyi causes a sexually transmitted disease called chancroid. The organism is very fragile. What you're going to see is a school of fishes morphology. So if you see that buzzword, associate that with Haemophilus ducreyi, which is a sexually transmitted disease causing chancroid. Bordetella. Bordetella pertussis is the cause of whooping cough. Bordetella peripertussis is the cause of kennel cough. If you get your dog vaccinated for kennel cough, they are vaccinating them against that type of pertussis, because the dog then will actually cough just like somebody who has whooping cough. How do you get it? You get it from droplet spray from other people. Okay, so it is passed from person to person. Now, Bordet-Gonneau media is the media of choice to use if you are growing it, and the organism looks like little mercury droplets. So if you see any of this, associate these buzzwords with Bordetella pertussis, which is the causative agent of whooping cough. Brucella. Brucella is an obligate intracellular parasite. It is a gram-negative rod. Brucella is associated with animals and bioterrorism. If you see a question about maybe a veterinarian who has a undulating fever. What does undulating fever means? Fever that goes up, fever that goes down. That could be associated with Brucella. So the thing with Brucella is that you must grow it a little bit longer on the media. It takes longer to grow. If you think you have it in a blood culture, you want to use a special blood culture bottle and incubate it longer. Legionnaires or Legionella. Legionella pneumophilia is the cause of Legionnaire's disease as we know it and was first identified. It is a waterborne organism that you'll see in water air conditioning towers. The organism needs a special ingredient cysteine in the media in order for it to grow. The best way to identify Legionella, because it is very difficult to grow, is serology. You can do a serological test, an immunofluorescent technique to identify it, and also an antibody titer on the patient. Well, you could probably do a brain biopsy after the patient is dead, but if you want to treat the patient beforehand, that might not be a good thing. Oh, that's true. Dr. House does brain biopsies. I've never heard of a brain biopsy in my on a live patient in my life. So, Campylobacter. What do we know about Campylobacter? It is microaerophilic. Remember, that term means that there is special atmosphere. Now, if you can't remember what atmosphere it is, remember that it is always higher nitrogen. Do you see this? High nitrogen levels. It is, think of Campylobacter as normal flora in chickens, and chickens are pretty hot. Their normal body temperature is 42 degrees, so you want to incubate Campylobacter at a high nitrogen, high temperature in order to isolate it better. Now remember, it can grow at 35 degrees, but you're using this temperature to isolate it from, say, fecal material. I noticed a lot of labs are doing now immunoassay techniques for Campylobacter because they can identify it much easier because they don't have to grow it. All right. It is oxidase positive. It has gull-winged morphology. So it is a curved gram-negative rod. If you see any of these buzzwords, associate it with Campylobacter. Helicobacter, Helicobacter pylori, is a gram-negative curved rod that is associated with ulcers. You can use a special breath test or biopsy. If you take a stomach biopsy and put it on a urea slant, you're going to get a positive urea test. That's how it used to be diagnosed. Now, of course, they use these breath biopsies, or they treat the patient with antibiotics and Pepto-Bismol for 1 month, and that usually gets rid of Helicobacter. Gardnerella vaginalis. Gardnerella vaginalis is associated with bacteria vaginosis. This organism will show those characteristic clue cells. All right, this is the one you can do the sniff test or the whiff test where you'll get a fish-like odor with 10% KOH. And remember, the presence of clue cells can be a clue to Gardnerella vaginalis. So think of clue cells and gardens. Now, let's talk about the Enterobacteriaceae, which is a huge group of organisms that are the gram-negative enteric rods. Now, remember some of the characteristics that go along with them and what we associate this group with. All right. One of the keys, too, is that they are all oxidase negative. If you have an organism that's oxidase positive, it is not in this group. The nitrate is reduced to nitrite. There are some exceptions, but remember on the urinalysis dipstick, we are using that characteristic because E. coli is the #1 cause of urinary tract infections. So we can actually see a positive test and associate it with this group of organisms. Enrichment media is used for stool culturing because we want to identify or be able to enrich organisms such as Salmonella and Shigella from all other normal flora in stool culture. Hectoin is another media that is used specifically for cultivation of stool, so it is direct inoculation of stool culturing. It contains lactose and sucrose and a pH indicator. Remember, the bright organisms are usually the lactose fermenters, and you're going to throw them away. In this particular instance, they turn salmon pink to bright orange. Your pathogens on most of the enteric medium are clear colorless or clear colorless with black dots. You guys remember that, right? I think I said it so many times that it is ingrained in my brain. I know that. Should be in yours too. We know what they look like. Remember, the bright colors are lactose fermenters. The clear and colorless, or clear colorless with the black dots, are our pathogens. Salmonella, Shigella, same thing. It is highly selective for these two organisms. H2S can also be detected. Bright colors, once again, show that they are the lactose fermenters. They are not the pathogen. Salmonella and Shigella are clear colorless or clear colorless with black dots. EMB is a media that is used in other parts of the country. Most of the people around here in these labs use McConkey, but EMB has lactose, and the thing about this particular plate is that E. coli will give you a green metallic sheen. So you can actually identify E. coli almost specifically with this EMB plate. Here it is. Notice the green kind of metallic sheen of E. coli. Now, triple sugar iron auger, what is it used for? To see if an organism ferments lactose, sucrose, and or all three. Where it is significant is if we see an organism that ferments glucose only, and that could be significant because that organism then could be an enteric pathogen. And remember, we're talking about stools here, or stool culturing. So acid over acid means the organism ferments lactose, sucrose, and or glucose. Here's some examples. Note that if it does nothing, you have alkaline over alkaline. You may have an organism such as Pseudomonas aeruginosa, which does not ferment. Remember, it has a special non-fermenting. It does it in a different manner. Now, your MVIC reactions are probably the easiest reactions to remember and easiest way for this group of organisms. We know what they mean. I is for indole, M is for Methyl-Red, V, Voges-Proskauer, and C is for citrate. So what organisms should you remember? Well, right away you should know E. coli is plus, plus, minus, minus, where Klebsiella is minus, minus, plus, plus. Those should be ingrained into your memory and into your flow chart, your basic flow chart for this group of organisms. Now, indole. What is the thing about indole? You must have tryptophan in the media in order for indole to be produced. Methyl-Red is a test that is not done except in the automated instrumentation anymore. VP, remember what is positive? It separates Klebsiella, Serratia, Anaerobic from E. coli, and from some Yersinia species that are also negative. Citrate separates your Klebsiella from your E. coli. Klebsiella is citrate positive. Question? Yes, they are. Remember, your MVIC test is in the little Vitek card or microscan card. All of those tests are on there. But remember, we're trying to remember these for testing purposes on our flowchart. Now, you should be able to look at, and I know a lot of people in the lab do this, they'll pick up their little Vitek card. They can actually see the reactions, or they'll go, oh, this looks positive, and this can't be a Klebsiella, or this is an unusual one if it is. Because they'll question some of those reactions that come up, because they do know these basic reactions. Now, remember, with lactose, you must have both of these enzymes in order for the organism to show lactose fermentation. the ONPG test detects for that, you must have permease and beta-galactosidase, because the lactose has to be brought into the cell and then it has to be utilized. So the ONPG test detects for beta-galactosidase. The PPA test, or phenylalanine deaminase test, we use this test to separate Proteus providentia and Morganella from all other groups. So remember, in your test question, if you see a PPA or a phenylalanine deaminase positive, you should be able to go immediately to your test questions and take E. coli and all others out of there. Deep carboxylase, and this is, some of you might remember this and some of you might not, so we're going to skip that slide. Urea. Urea can also be used in your flow chart, because remember, rapidly urea positive is your Proteus providentia morganella. Your urea positive in 24 hours are your Klebsiella anaerobacter serratia, and urea negative then are your E. coli, Salmonella, Shigella on your far, if we use the flow chart, it was on the far right of our flow chart. Vibrios. Vibrios then are not in the Enterobacteriaceae group. This is in the Vibrioaceae group. Vibrios are curved gram-negative rods. The Vibrio cholera is associated with the buzzword rice water stool. We had a question previously of that. Pardon me? Yes, the bed, the cholera bed, because you have massive amounts of diarrhea, and that is rice water stools. Parahemolyticus and vulnificus can cause gastroenteritis. Now, blue-green colonies on TCBS are a positive test for Vibrio cholera. Usually not only do you see this mentioned, you might also see mentioned that it says rice water stool. So even if you didn't know TCBS, you should be able to pick cholera. All right, the non-fermenters are a group of organisms that do not use oxygen in the normal way that, say for our Enterobacteriaceae group. So they don't ferment, but they oxidize. So in this particular case, you want to use special media, and no, you inoculate this media by taking a stick and going right down into the bottom. An organism that can ferment on the top and on the bottom, this is a fermenter. This organism right here shows it can do it on the top where there's oxygen, but it cannot utilize any of those carbohydrates where there is no oxygen. So that puts those organisms in this group. Many of these organisms are what we call the bench top, or organisms that are contamination or sink top organisms. The most common in this group is Pseudomonas aeruginosa. You should know this organism. It has a grape-like, grape soda smell or a corn tortilla smell. It is oxidase positive, and it has a green pigment. And it is also very antibiotic resistant. All right. Stentrophomonas multifilia used to be called Pseudomonas multifilia. This is an oxidase negative Pseudomonas species that I say. It is also commonly identified along with Acinetobacter and Moraxella. Now, one of the characteristics too is do an oxidase test. All right, let's briefly talk about anaerobes. Question? Pseudomonas, how pathogenic is it? You can have different strains of it. Some of them have capsules that can be more pathogenic. It tends to be more pathogenic in immunocompromised patients than not. A lot of people, since we deal with it on a day-to-day basis, it doesn't bother us. But say if you swim in a pool or somewhere that they don't properly bleach it or, decontaminate it. You can get it down in your ears, and kids can get it a swimmer's ear and those kind of things. But it, very common in wound abscesses or contamination in ORs because it is so resistant to antibiotics and disinfectants. That's the problem with it. It is one of what we call the sink top contaminants. Like these types of organisms were thought to be non-pathogenic because you could just culture them from your sink. And then what they found was they were crossing over into the more immunocompromised patients. And then growing in the iodine. If you remember that story I told you about, the Pseudomonas actually grew in the iodine in the OR. So most of the time these organisms are contaminants, and then they are contaminants that cross over as infections. So Pseudomonas aeruginosa is the most commonly identified cause of infections in that group, in that non-fermenting group, along with a few others. Okay, but that is the most important one to know. And remember, it is oxidase positive. So it is not in the Enterobacteriaceae group. It's in a different group. Now your anaerobes, you want to process a little bit differently because you must put them in an anaerobic jar. They should be then incubated so that that anaerobic jar takes out all of the oxygen in that jar, in that atmosphere, so the organism can grow. Now methylene blue is used as an indicator. That is what is on the strip here. So methylene blue is blue in oxygen, and it is colorless without oxygen. That might be a question they ask, what is the indicator, or what color should the indicator be? Or they might give you an indicator color, and then you have to troubleshoot. So anaerobic media is media that does contain antibiotics, etc., because you might want to be going from an area of high normal flora, and you want to be able to culture out the obligate anaerobe. Aerotolerance we talked about earlier, you know what that is. Now your anaerobic gram-negative rods are the group of Bacteroides fragilis group. They are biopositive and many are pigmented. This is one of the most common gram-negative anaerobic rods that causes infection. Your anaerobic gram-positive rods are things like Bacterium-acnes. This is the one that's on people's skin that can cause skin acne, and you can also contaminate your blood cultures with it. Actinomycetes, as we said earlier, is your sulfur-containing organism, or your sulfur granules. Now, your anaerobic gram-positive rod spore-forming, we talked about earlier as being the Clostridium. Clostridium perfringens, the buzzword that goes along with it is the double zone of hemolysis. You're definitely going to see that in a test question, and refer to it back as perfringens. That is the cause of gas gangrene. All right. Clostridium botulinum, food poisoning, very potent neurotoxin, Botox. Clostridium tetani, you see the terminal spores that are either called drumsticks or lollipop, cause of lockjaw. Your Clostridium difficile, which we use the C. diff test. What are we testing for? We're testing for the toxin and the antigen. So the antigen is the actual organism, and we're also testing for the different toxin. The reason we do this is because it is very difficult to grow because it is so sensitive to oxygen. But you're going to see in the test question pseudomembranous colitis induced by antibiotic use. So you're going to see this in patients who have been treated with big gun antibiotics. Mycoplasma pneumoniae, cause of walking pneumonia. You can also get a cold agglutinin with Mycoplasma pneumoniae. If you grow it, you'll get fried egg colonies. This group of organisms has no cell wall, so you have to use special media to grow it. Rickettsia. Rickettsia is a obligate intracellular parasite. It is a gram-negative Coccobacilli. Remember the story about Lyme disease, that Gary Bergdorf-Ferry was working with Rickettsia because one of the Rickettsia organisms causes Rocky Mountain spotted fever. So many of the organisms in this group are passed by an insect vector. They are very pathogenic. Now, we used to do what is called the F fever of unknown origin test. I don't think they even ask you any questions about that anymore. But what they did was they used Proteus strains, which they called OX strains, because they immunologically cross-reacted with antibodies to Rickettsia. So you didn't have to expose yourself to Rickettsia. So they are like the OX19, OX20. Those are the cross-reacting antibodies. Chlamydia is an obligate intracellular parasite. Chlamydia psittaci is the one that causes psittacosis in birds. Chlamydia trachomatis is the one that causes the non-gynococcal urethritis in humans. We now use PCR as standard testing for chlamydia. Most of the time we're doing PCR for GC, gonorrhea, and chlamydia. Borrelia burgdorferi, we mentioned that last week when we talked about immunology. The bullseye rash transmitted by the deer tick, Ixodes domini. Now, briefly, let's talk about antibiotic sensitivities and what you need to know. What is the MIC test? MIC, KEYMOUSC. So MICs are now broth dilutions. They are routinely done on your Vitek, your Microscan, also along with disc diffusion, which is non-automated. Remember that MICs give you the highest dilution, or excuse me, the lowest concentration of agent in which the growth is inhibited. And then that is associated with the concentration that you can achieve in the patient. So if the MIC, if that concentration can be achieved in the patient, then it is said to be sensitive. If it can't be achieved in the patient, it is said to be resistant. Remember that the Kirby-Bauer is then associated with MICs. So remember, with MICs, your organism concentration should be your McFarland standard of 1. And the McFarland standard of 1 should not be used for your Kirby-Bauer disc diffusion, because that's too much organism and it can affect your disc diffusion test. Now, your disc diffusion test, a couple things to remember is that you want to make sure that you use a 0.5 McFarland standard. The 1 standard means more organism. 0.5 is less organism. Remember all of the things that we talked about with the depth of the auger, the concentration of the disc, and how the outcome of the test will be skewed if you do not use the appropriate media, the appropriate type of disc, the incubation time, and the temperature. All right, discs have don't place any more than 12 per plate, and then you want to measure the zone of inhibition. Now that zone of inhibition, you have to go back to a chart. Then that chart associates that zone of inhibition back to an MIC. Now, sometimes the needs of a fastidious organism, you need to add blood to your Mueller-Hinton. You can add sheep blood to the Mueller-Hinton agar and do a disc diffusion test. Now, make sure during quality control that all of those zones are recorded as part of your quality control with known organisms. The E-test is also done. E-test is just done a little bit differently because you're measuring kind of this teardrop area zone. All right, and this can be done on organisms that are more fastidious, for instance, on a Mueller-Hinton plate with blood. Now, what is the McFarland standard? It is barium chloride. The barium chloride is diluted to give you an optical density or an absorbance. Remember, optical density and absorbance are the same, of 0.5 and or 1. Many of you that did the Vitek testing in the lab, you put your little tube inside this tester and push the button, and then you had a little dial that went over like into the green zone, or if it was in the red zone, it meant it was too concentrated. You added some saline. If it wasn't enough, you let it grow for a little bit longer before you actually inoculated it. Now, methicillin-resistant staph, we know we test the oxacillin disc to see if you have methicillin-resistant Staph aureus. Your antibiograms, if you have a MRSA, I don't know that. The question was, is that considered a critical value? So I guess if the patient has never been identified as having MRSA and you identify it as an oxacillin-resistant species, you want to make sure the patient is isolated. So I guess, yes. Antibiograms, the one that is significant is your Klebsiella, because you have carbenicillin and tricaricillin resistancy. I think I read just previously that there are some that are showing a little bit different patternings, okay? But as a rule, most techs think with Klebsiella that these are still accurate. Now, QC, remember with QC in microbiology, always use your biological safety cabinet, use universal precaution, make sure your media is stored, logged, and dated at the appropriate temperature. Make sure you choose an appropriate battery of tests for that appropriate type of organism. Discard anything past its expiration date. Don't use it. Don't say it's okay for this particular time. Make sure you run positive and negative controls for all reagents, all stains. Some of you that are going to microbiology right now know that you need to make sure that you say, well, did you run the controls for that particular reagent? Did you run the controls for the catalase test today? Let me run the controls along with my test. Let me run the controls for the oxidase test. All right, those things are done daily. You should also monitor your temperature of the equipment and your lab environment. What is the temperature of the room you're in? Because that is important. All right, any questions about microbiology?